Hello everyone, in this class we'll try to learn about synapse and what are the different properties of synapse. Synapse. Information about external environment is sensed by receptors which acts as a transducers. They convert sensation to action potential which travel via nerve fibers to the center. Hence, there is a series of nerve fiber to the center. The junction between different neuron is known as synapse. It may be a junction between two neurons, neuron and a muscle, neuron with the different glands of the body. There are different types of synapses. They are exodendritic, exosomatic, exoaxonal. Exodendritic where axon of one neuron synapse with a dendrite of other. Exosomatic is where axon of one neuron synapses with a soma of other. Exoaxonal is where one axon is going to synapse with another axon of a nerve fiber. So this diagram shows different types of synapse: exodendritic, exosomatic and exoaxonal. Physiologically, the synapses are classified into chemical synapse and electrical synapse. Chemical synapses where the transmission of impulse from pre to postsynaptic site occurs due to the release of chemical mediators, whereas gap junctions which are formed by the close approximation of pre and postsynaptic membranes acts as a low resistance bridges in electrical synapses. Chemical synapses are seen in most of the synaptic junction in the nervous system. They are unidirectional from presynaptic to postsynaptic, whereas electrical synapses they are very few in the central nervous system, and they can transmit signals in either direction. For example, lateral vestibular nucleus, cerebral cortex, and in hippocampus they are present. There is a presence of synaptic cleft in chemical synapse, whereas synaptic cleft is replaced by resistance bridges in electrical synapse. Synaptic delay is present in chemical synapse, whereas it is absent due to the low resistance bridges. Chemical synapses they are sensitive to hypoxia, where electricals they are insensitive, and infinite processing of information is possible in chemical synapse, whereas transmission is rapid but efficient, but there are not too many synapses in electrical synapses. Electrical events that occur at synapses. Single stimulus applied to the sensory nerves does not lead to the formation of propagated action potential. In postsynaptic neuron, rather it produces transient partial depolarization or transient hyperpolarization, which is called as EPSP or IPSP, whether they are excitatory or inhibitory. Excitatory postsynaptic potentials. The combination of chemical transmitters like glutamate and aspartate with the receptors make the postsynaptic membrane partially depolarized. That is, there is a drop of potential across the membrane from, for example, minus 80 to minus 65 millivolts. This difference of plus 15 millivolts is called as excitatory postsynaptic potential, that is EPSP. The initial depolarizing response produced by single stimulus to the sensory input begins at 0.5 milliseconds after the afferent impulse enters the spinal cord, and then it reaches at its peak at 1 to 1.5 milliseconds later, then declines exponentially over the next 4 milliseconds if not sufficient to cause generation of the spike. During this period, the excitability of the neuron to the other stimuli is increased, and consequently the potential is called as EPSP. The excitatory postsynaptic potential makes the postsynaptic membrane hyperpolarized, and if sufficiently strong, it causes action potential to develop in the postsynaptic membrane. Ionic basis of development of EPSP. When action potential arises in the presynaptic knob, the synaptic neurotransmitter is released and it combines with the receptor of the postsynaptic membrane, and this combination causes opening of sodium or calcium ion channels in the postsynaptic membrane. The entry of sodium or other cations inside the cell from the extracellular fluid leads to hyperpolarization and development of EPSP in the postsynaptic membrane. EPSP can also be produced by agents that close potassium channels. Excitatory postsynaptic potential due to activity in one synaptic knob is small, but EPSPs produced by each of the active knob can summate, and this summation may be spatial or uh, temporal summation. Spatial summation. On stimulation of two afferent fibers simultaneously, both which make synaptic connections with the motor neuron, their APSPs get summated to the critical level, and thus they may produce spike discharge from the motor neuron, and this is called as spatial summation. In other words, the activity in one synaptic knob facilitates the activity in another to approach firing level. Temporal summation. If we stimulate an afferent nerve for the second time before the decay of first stimulation, neither of which alone can generate a spike from the motor neuron. However, each produces some EPSP which gets summated to the critical level necessary for the initiation of spike potential, and this is called as temporal summation. This is diagram showing the temporal summation and the spatial summation and the temporal summation. So here at the postsynaptic membrane, two different stimuli they produce different EPSPs, and they are going to summate to produce action potential, whereas temporal summation at one postsynaptic membrane, the at the single spot there will be multiple stimuli one after another. They are going to produce EPSPs, and these EPSPs they are going to summate, and if the potential it is going to reach to the threshold stimulus, then it is going to produce action potential. Inhibitory postsynaptic potential. Sometimes the combination of chemical transmitter like GABA or glycine in the brainstem and spinal cord with the receptors make the postsynaptic membrane hyperpolarized. For example, resting membrane potential is going to reach minus 90 from minus 80, and this difference of minus 10 millivolts is called as inhibitory postsynaptic potential or IPSP. Hyperpolarization begins at 1 to 1.5 milliseconds after the entry of afferent impulse into the spinal cord and rises to peak in 1 to 5 milliseconds after its onset, and declines exponentially with the time constant for about 3 milliseconds. During this potential, the excitability of the neuron to the other stimuli is decreased, and consequently it is called as inhibitory postsynaptic potential. Ionic basis of development of IPSP, inhibitory postsynaptic potential, is due to inhibitory chemical transmitter liberated on the subsynaptic membrane. Therefore, it becomes the membrane becomes permeable for potassium and chloride, but retains high degree of impermeability to the sodium. This results in hyperpolarization of the whole postsynaptic membrane. IPSPs can also be produced by opening of potassium channels with the movement of potassium out of the postsynaptic cell. In addition, they can also be produced by closure of sodium and calcium channel. This is diagram showing of the IPSP, that is inhibitory postsynaptic potential, where sometimes membrane potential can reduce from minus 40 to minus 50. Coming to synaptic inhibition, there are two types of synaptic inhibition: postsynaptic inhibition and presynaptic inhibition. Postsynaptic inhibition, it depends upon the delivery of an inhibitory chemical by the incoming axons which hyperpolarize the subsynaptic membrane. It is subdivided into direct or indirect inhibition. Direct inhibition of the postsynaptic membranes occurs by development of the EPSP. For example, afferent fibers from the muscle spindle in skeletal muscles are known to pass directly to the spinal motor neuron of the motor unit supplying the same nerve in a stretch reflex. Impulses in this afferent supply causes EPSP, which summate to produce propagated response in postsynaptic motor neuron. But at the same time, IPSPs are produced in the motor neurons supplying the antagonist muscles. This latter response is mediated by branches of afferent fibers that end on Golgi bottle neurons or interneurons. So these interneurons in turn secrete inhibitory transmitter, that is glycine, at the synapses on the proximal dendrites of the cell bodies of the motor neurons that supply the antagonist, and this is example for direct inhibition. Therefore, activity in the afferent fibers from the muscle spindles excite motor neurons supplying the muscles from which the impulses come and inhibits those supplying its antagonist, and this reflex is called as reciprocal innervation. There can also be indirect inhibition of the postsynaptic membrane. This can occur because of the effect of previous postsynaptic neuron discharges. For example, the postsynaptic membrane can be in a refractory phase, and it can be refractory to the excitation because it has just fired and it is in the refractory period. It can also occur due to after hyperpolarization, where the membrane potential becomes more negative, and it can also seen in Renshaw cell inhibition or negative feedback mechanism, which is also called as a negative feedback mechanism. Okay. So this diagram showing the postsynaptic inhibition here. The postsynaptic inhibition can be direct when the interneuron secretes inhibitory neurotransmitter, or postsynaptic inhibition can occur because of the effect on the postsynaptic membrane, whether when it is there in the refractory period. Okay, so then the cell is not cannot be excitable. Renshaw cell inhibition. In this condition, motor nerve gives off some collateral branches as they traverse the spinal cord towards the ventral root, and these collaterals make excitatory synaptic connections with interneurons called Renshaw cell, situated in the ventromedial part of the ventral horn. These Renshaw cell in turn send axons which make inhibitory synaptic connections with the motor neurons, and Renshaw cells are stimulated by acetylcholine. So this diagram showing the Renshaw cell inhibition, where you can see the anterior motor neuron. So when it discharges, it sends some collaterals to the Renshaw cells which are inhibitory in nature. They, by negative feedback mechanism, they can inhibit the other motor neurons by producing negative neurotransmitter, and these cells becomes inexcitatory to the other impulses. Okay? And this is example for the postsynaptic inhibition. Presynaptic inhibition. This type of inhibition occurs at the presynaptic terminals before the signal ever reaches the synapse. Presynaptic inhibition occurs because of the failure of the release of excitatory neurotransmitter substance from the presynaptic axon terminal. This occurs in synapses where inhibitory neuron synapses with the afferent fibers of excitatory neuron before the later synapses with the efferent neuron. In other words, presynaptic inhibition occurs because of the axo-axonal synapses. Okay, so this is the diagram showing the presynaptic inhibition. Here, this is usually the excitatory, and this interneuron it forms the axo-axonic synapse with this excitatory neuron, which is usually inhibitory. It secretes inhibitory neurotransmitter, thus it is going to suppress the excitatory impulse on the motor neuron. Activation of presynaptic receptors reduces the opening of calcium channels directly, or increase chloride and potassium conductance, so which indirectly causes or reduces the calcium entry, and this decrease of the size of the action potential reaching the excitatory ending. So the amount of excitatory transmitter released is reduced. The inhibitory transmitter at the axonal synapse is usually GABA. Acting via GABA A receptors, it increases chloride conductance, whereas GABA A receptors via G protein produces an increase in potassium conductance. Other transmitters also mediate presynaptic inhibition by G protein mediated effects of calcium on uh, G protein mediated effects on calcium channels and as well as potassium channels. Note that there is also a phenomenon called as presynaptic facilitation, which is produced when action potential is prolonged and action and calcium channels are open for longer period. Therefore, repolarization gets delayed and allowing the repolarization to continue. It is mediated by serotonin. Serotonin released at the exoaxonal endings, it increases intraneuronal cyclic AMP, and this results in phosphorylation of one group of uh, potassium channels, closes the channels and slowing the repolarization and prolonging the action potential. There is also a phenomenon called as feed-forward inhibition. It is seen in the cerebellum. In this type of inhibition, a neuron is connected through two pathways. One is excitatory and another is inhibitory. For example, in cerebellum, stimulation of basket cells produces iPSP in Purkinje cells. However, the basket cells and Purkinje cells are excited by the same parallel excitatory input, and this arrangement is called as feed-forward inhibition. This helps to prevent the duration of the excitation produced by any given afferent impulse. Okay, so you can see uh, the the basket cells, I mean, they are mainly excited to the inhibited to the Purkinje cells, but some of the afferents which are going from the basket cells, they themselves stimulates the Purkinje's fibers. And the basket cells usually it modulates the excitatory output from the Purkinje cells, and this kind of inhibition is called as feed forward inhibition, which is seen in cerebellum. Coming to the properties of synapse, some of the properties are convergence and divergence. Many presynaptic neurons meet on a common focus on any single postsynaptic membrane called convergence. For example, in spinal motor neuron, some inputs from uh, some inputs come from directly the dorsal root, some from the long descending spinal tracts, and many from interneurons. The axon of the most presynaptic neurons divide into many branches that separate out in different direction to end on many postsynaptic neurons is called as divergence. One-way conduction is another property. The synapses permits conduction of impulses in one direction only, that is from presynaptic to postsynaptic neuron. This is because the chemical mediator at the synaptic junction is located in the synaptic knob of the presynaptic fibers but not in the postsynaptic membrane. And another property is synaptic delay. When an impulse reaches the presynaptic terminus, there is a gap of about 0.5 milliseconds before a response is obtained in the postsynaptic neuron, which is called as synaptic delay. This is due to the time taken for synaptic mediator to be released and to act on the postsynaptic membrane. And its significance is the conduction along a chain of neurons is slow if there were many synapses in the brain. Okay, and it is possible to determine whether the given synapse is monosynaptic or polysynaptic. Property is synaptic fatigue or habituation. Here, repeated stimulation of presynaptic neuron lead to gradual decrease and finally disappearance of postsynaptic response. For example, in epilepsy. The fifth property is summation, where the hypothetic nerves neuron A and B converge on neuron X and B diverges on X and Y. A stimulus applied to A or B will set up an EPSP in neuron X. Here you can see the stimulation of A and B is going to set up action potential in X, the property of which is called as convergence. And or it is also a partial summation. And when the neuron B is stimulated repeatedly one after another, it is going to stimulate X and Y to produce action potential in them, and this property is called as divergence, and also it is an example of the temporal summation. So here, if A and B are stimulated at the same time, two areas of depolarization will be produced in X, and their action will sum up is called as partial summation. If the neuron B is stimulated repeatedly, X and Y will discharge, and it is called as temporal summation. Another property is occlusion. When two afferent nerves, A, which is supposed to be having 5 subunits, and B, which is again having 5 units, to a skeletal muscles are simultaneously stimulated, it is sometimes seen that the tension developed by the muscle under observation is less than the sum of tension produced by each afferent nerve stimulated separately, and this phenomenon is called as occlusion. This is because some of the motor neurons, which are common to both, are excited maximally when either A or B is stimulated separately. They give no greater response when A or B stimulated together. This decrease in response is due to the presynaptic fibers sharing postsynaptic neurons. Thus, the occlusion is due to the afferent fibers overlapping in their central distribution. Okay, so here this diagram shows that when A and B, they are separately stimulated, they will be able to excite 5 neurons, and when they are stimulated together, they will be able to excite only 8 neurons, because some of the neurons, they are common to both A and B, and this property is called as occlusion. Subliminal fringe. Subliminal means below the threshold, and fringe means border. Thus, the postsynaptic neurons are said to be in subliminal fringe if they are not discharged by activity in the presynaptic neurons, but do have their excitability increased. When two afferent excitatory nerves, that is nerve A, which is supposed to excite 3 units, and B, which is supposed to excite 3 units to a skeletal muscles, are stimulated at the same time, two areas of depolarization will be produced, and sometimes the tension developed by the muscle is greater than the sum of the tension produced by each afferent nerve stimulated separately. For example, when the neurons A and B stimulated simultaneously, they will be able to produce excitation in 3 neurons. But when they are stimulated simultaneously, they will be able to excite 8 neurons, which is more than 3 plus 3, that is 6. This occurs because there are some common neurons which are in subliminal fringe. So this occurs because effect of each depolarization while fully activating a certain number of motor neurons also acts on further number subliminally, and that some of these subliminally influenced motor neurons are common to A and B. As a result, subliminal excitation can thus sum up to produce threshold stimulation. So this is another example of spatial summation. The eighth property is synaptic plasticity. Plasticity means capability of being easily modulated or changed. Synaptic conduction thus can be increased, that is strengthened, or decreased or weakened on the basis of past experience. These changes can be presynaptic or postsynaptic and play an important role in learning and memory. Some of the forms of synaptic plasticity. Post-tetanic potentiation. Here, a brief tetanizing stimuli in the presynaptic neuron results in increased production of postsynaptic potential lasting up to 60 seconds. It occurs due to the increased calcium in the presynaptic neuron, and hence the response is potentiated. Long-term potentiation. If post-tetanic potentiations get much more prolonged and last for days, it is called long-term potentiation. It is due to increase in the intracellular calcium in the postsynaptic neuron rather than presynaptic. It occurs in many parts of the nervous system, but it has been studied in greatest detail in hippocampus. There is also long-term depression. It is opposite to the long-term potentiation. It resembles LTP in many ways, but it is characterized by decrease in synaptic strength. It is produced by slower stimulation of presynaptic neurons and is associated with smaller rise in intracellular calcium than occurs in LTP. This phenomenon is commonly occurs in hippocampus and cerebellum. Another synaptic property is habituation. When the stimulus occurs again and again, the response to stimuli decreases. It is referred to habituation. Sensitization. It is the prolonged occurrence of increased postsynaptic responses after a stimulus to which an animal is habituated is paired once or several times with a noxious stimulus. It is due to presynaptic facilitation. Sensitization may occur as a transient response or if it is reinforced by additional pairings of noxious stimulus and the initial stimulus. It is mainly due to the calcium-mediated change in the adenyl cyclase that leads to greater production of cyclic AMP. So this diagram shows the site of the post-tetanic potentiation, the fatigue or habituation, which occurs mainly in the presynaptic membrane, where with the post-tetanic potentiation there will be increase in the calcium levels. And sensitization occurs because of the presynaptic facilitation, whereas long-term potentiation occurs because of the increase in the calcium levels, whereas long-term depression occurs because of the slow release of calcium or low levels of calcium in the postsynaptic membrane. There will be the pH also is going to affect, where alkalosis when pH increases, the neuronal excitability is markedly elevated. When in acidosis, where pH decreases, the neuronal excitability is decreased. For example, in diabetic ketoacidosis, neuronal excitability is going to decrease. So this is in brief about synapse and different properties of synapse. Thank you.